HBV antibody GIGA 2339 approved by FDA for clinical trials for hepatitis B cure. The new therapy, called GIGA 2339, is now set to enter Phase 1 clinical trials by following a recent FDA approval of the company's investigational new drug application. GIGA 2339 is developed by gigagen, a grifols company. GIGA 2339 is the first recombinant human polyclonal antibody treatment for HBV, and the FDA approved this drug to begin trials in late 2024. Carter Keller, the senior VP at Grifols and head of gigagen. He said, "There has been significant progress in the development of the first recombinant human polyclonal antibody therapies aimed at a functional cure for hepatitis B virus, HBV." Keller stated, "It consists of thousands of antibodies all directed towards the hepatitis B virus. We call these types of therapies recombinant polyclonal antibody therapies, which are very unique." He also said, "What we find with our diverse antibody therapy is that it's over 2,000 times more potent than currently available therapies, as demonstrated in both in vitro and in vivo experiments." In preclinical studies, GIGA 2339 showed more than 2,000 times the potency of current plasma-derived HBV treatments, suggesting it could effectively neutralize and clear the virus. This unique therapy could potentially provide a functional cure for HBV and can be scaled efficiently in a lab setting. Keller outlined the objectives and endpoints. For Phase 1 trials, the primary outcomes typically focus on safety and tolerability, while pharmacokinetics are secondary outcomes. What's exciting about our upcoming trials is that we have both a single ascending dose and a multi-ascending dose study. The FDA has agreed that the right patient population for this study are those with chronic hepatitis B infection. We will be exploring various endpoints, including markers of chronic infection, levels of the virus, and surface antigen levels to assess how potent we are in humans at different dose levels, Keller explains. Currently, there is no cure for HBV, but existing treatments offer only minimal reductions in HBV protein levels, leaving patients vulnerable to conditions such as hepatocellular carcinoma and cirrhosis. GIGA 2339 aims to address this gap by using more than 1,000 anti-HBV antibodies, replicated from the natural immune response of vaccinated donors. Keller discussed the scalable nature of GIGA 2339's production in a lab setting, stating, "One of the traditional methods for producing hepatitis B treatments involves extracting from human plasma, which is a limited resource. To create a potent hepatitis B drug, you need to continuously vaccinate patients, collect their plasma, and then turn it into a drug. By producing GIGA 2339 in the lab at scalable levels, we can meet global demand without relying on patient-derived plasma. If the drug proves to be as potent as expected, we can produce enough to treat the 300 million patients currently suffering from chronic hepatitis B." The development process began with blood from HBV-vaccinated donors, from which gigagen isolated antibody-producing B cells. These cells were used to extract antibody-coding DNA and create cell lines that produced synthetic human antibodies targeting HBV. We captured immune systems from people who've been vaccinated with the hepatitis B vaccine, and we took the best immune responses, ran those through our platform, and filtered out the antibodies focused just on hepatitis B surface antigen. He said, "We believe it has a real chance to cure HBV." Keller concludes, "I am very excited for the trials. Creating thousands of antibodies against a specific disease recombinantly was once considered impossible, and we're thrilled to show that not only is it possible, but it's also highly effective in patient populations." If you like this video, please like and share this video more and more. Please support our channel. Thanks for watching Great Wisdom Discover. We will meet you with new update in the next video. Thanks for watching Health Wisdom.